everyone. My name is Sanjana, a third year engineering physics student at IIT Roorkee. Today, I'll be explaining the application of AI to detect addiction patterns in brain imaging. In this video, I'll talk about understanding the problem statement, uh, having a research paper discussion, forming clear objective of this project, discussing a layman logic that we'll try to implement, and a code review for the same, and finally a conclusion for our project. So let us begin with the problem statement itself. It says applying AI to detect addiction patterns in brain imaging. So there can be a lot of questions in mind. Such kind of a problem statement is very vague. You can have questions about how is brain imaging done, or it can be like what visible changes our brain shows when it is exposed to drugs. What are addiction patterns exactly? What are the different regions of the brain? Uh, you might not even know about the effects of drug, or you might have the lack of biological understanding in general. And this is a very common issue that AI ML coders face, is that they might not have prior knowledge about the field they are trying to use this AI ML in. So for this, research is very important. Research, understanding basic terminologies is very important, and then finally we can understand where will we apply this AI ML knowledge in. So, moving forward with a research paper discussion that will give us a clear concept of what is our problem statement. So, I think we all know about X-rays. X-rays is done by using electromagnetic radiation. That's X-ray itself. For magnetic MRI, magnetic resonance imaging, we make use of a magnetic field, and in this, we measure how water molecules interact in such kind of field. Our body is 70% water. All the organs, the tissues in our body are primarily constituent of water. So that itself provides a good base for MRI to be used in. And MRI produces detailed images of such organs, tissues, white or gray matter in our brain. This MRI now for our data analysis is stored in a compressed format in this .NIIGX file, such as like a .PDF file. And and MRI is actually a 3D imaging. So what happens is that there are slices of 2D images that is prepared. And when these 2D images are clubbed together, we get a 3D image. Then 2D images have something called as a pixel. A 3D image has a volume pixel, a pixel which has volume associated with it. So to give a better understanding of this, a pixel is flat, while a voxel is a 3D pixel. Each and every voxel of a corresponding MRI image has predefined volume. It has a predefined length, it has a predefined breadth, a predefined height, and therefore a predefined volume. Such volume is not generalized. Each and every image has its own volume, though there are standardized volume that exists. So another question can be, how does drug exactly affect our brain? So what happens is that when when someone intakes drug, it actually releases dopamine, and dopamine is our like happy hormone, so it causes this immense pleasure, and that is why uh, people fall into such kind of addictions. They more frequently use it to get such dopamine hit. There's a particular region in our brain, and that is called the nucleus accumbens, NACC. It is a critical part of our brain's reward system, and this will be our project's primary target, so we'll keep this in mind. So there is um, a group in the University of Belmont. It is called as the Enigma Addiction Working Group. So they hold a lot of researches, and they release their research findings. One of such research papers I found out to be very helpful It's said that the NECC is very much related to addiction. This paper suggested that the volume asymmetry is altered in substance dependence. So we know our brain has a left hemisphere and a right hemisphere, but the hemispheres are not same. Yes, there is asymmetry in a normal human being too. But the paper said that the addicted individuals had less leftward asymmetry. So, yes, we have asymmetry, but the addicted individuals had less asymmetry. To keep this in mind is very important. So the Enigma Addiction Working Group does a lot of research not only on drug abuse but also on alcohol and alcohol with drug abuse, and they have several research papers on this. Though their data access is very limited for public, it is very confidential and not for the use of public, so I could not get hands-on data access, which would have been wonderful. Uh, but with this project, we will make use of their research paper, which was very understanding, and we'll see in our project if their research paper uh, has some right conclusions, whether I'm able to match it on some other data set, which is not theirs. So, after learning the basic terminologies, we can now easily define our objective. It is simply measuring the volume of the left and the right NACCs in MRIs using AI, calculating their asymmetric index, and checking this brain pattern in drug addicted person. So, what is asymmetric index? I'll tell further, but it is basically checking the asymmetry between the left and right NACCs, and we want to see if yes, definitely there is less asymmetry in drug addicted person or not. We want to see this. Uh, so now we have our we have our objective defined. Moving forward for the logic, even if we have these brain images, these MRI images, we don't know which region corresponds to what. We are not a surgeon, we don't have biology, we're BTech students. So how would we know which region corresponds to what? So we will make use of a tool called ANTS, and for short, it is an advanced normalization tool. Now, this is a very heavy term, so let me give you a very real world analogy. Let us say your teacher has given you a India map. In this India map, she asked you to label all the states. Now, it is manually very difficult to do because the lines are not symmetric, the lines are not perfect, they are not perfect circles, there are indentations. So, how will you do it? What you'll do is you'll take a sample map, you will put it on top of your India map, and then you will outline it, and then you will get your labels. Perfect. See, this is how you can do it. So we'll use the same concept in brain MRI imaging. So what happens is that we have a normal MRI image and we'll take a standard image, a reference image. This reference image, we'll take, this reference image is called an atlas, and we'll use the Harvard or Oxford Atlas, which contains these predefined labels for various different brain regions. And then we'll put it on top of, we'll wrap it around the brain, and then we'll get these regions labeled. But the thing is, it might happen that a brain image is smaller. It might happen that maybe it is rotated slightly. So in such case, we use affine registration, which is nothing. It does rotation, translation, scaling, sharing means cropping it to perfectly align both of them together. And AND uses this register. And ANDS is a tool to wrap this atlas around. So what happens? ANDS helps us to wrap this atlas around and affine registration adjusts it. To understand this atlas better, there's a small code snippet. I have written print atlas and it has given me a lot of labels. There's a left cerebral white matter, and similarly, there might be a right cerebral white matter. So yes, there are left and right labels given. And in computer language, we have IDs for this. So each vertex, so we have discussed vertex before, it is a 3D pixel. Each and every 3D pixel gets its own label. So let us say the left cerebral white matter, they get a vertex of 1 according to this. So anywhere there is a left cerebral white matter, they get a Bolex of 1. So uh, for our project, we want to focus on the left NACC and the right NACC, and we do that by taking the index 11 and 21. I have done this manually. Moving on to the next part is defining the asymmetric index. Asymmetric index is nothing but the difference in the left NACC volume and the right NACC volume. And I've taken a ratio so that I'm able to compare it with all the participants in my data set. There's a small code snippet over here where I've written the left volume is equal to, I've taken only the vertex, the 3D pixel with having index 11 for the left NACC. I've summed, so I've gotten the total number of NACCs, the left NACCs, and I've multiplied it by the vertex volume. This vertex volume I'll move forward in the code to explain how I got it, but it is nothing but length, breadth into height. That's it. Finally, we get an our asymmetric index. This asymmetric index I'll calculate for all the patients I have, whether it be a healthy control or a drug addicted patient. We'll compare the results for both of these. So now let us move on to the core part of it, data set. So I have made use of OpenNeuro. Like Kaggle offers various free data sets. OpenNeuro is only for medical data set. It's just for medical research purposes. And it has all sort of data sets from be it MRI or be it any X-ray scan. You can get anything over here. You have to use specific keywords. And I found this Mexican data set of cocaine use disorder patients. We call them CUD patients. I found the data set for the CUD patients. Over here, it contains their license, readme, and other stuff. But the most important is the 161 participants. So this is very important. It has 160 participants. Each participant has some number of files. They're not equal. But our main aim is to look at the MRI file, and it is located at the NAT file. We can see this is our MRI document in which we'll be performing our research on. Every subject has an NAT file at least. So we'll make use of this MRI image to see the volume asymmetry. Because such data set is a very heavy, we make use of Amazon Cloud to download it. Manually downloading it from OpenNeuro does not work. So OpenNeuro provides uh, a code to download it from Amazon. They have very specific number key given to each and every data set. For this, it is DS003346, and you can check it out yourself when you go into OpenView. If you type this, you'll get the data set automatically. We'll make use of Amazon Cloud. First, we'll install Amazon Cloud, then we'll define the path where we want to download it, and then finally we'll download the OpenView data set. Then we'll move on to labeling different regions of the brain. Now, since I'm doing this for 161 participants, the loop might become a little tricky to understand. We would want to look at one participant folder at a time. So we'll do that first to understand the logic we are trying to implement, and then we'll look for the 166 participants and how I've used the loop to calculate for all the 161 participants I have. So this is like my test sheet. In this, again, I've loaded all the 161 participants. I've downloaded all the important libraries. I've imported all the important libraries. So let us again look at the data set. This is the one that I was showing in OpenEuro. Same, I have exported it here as well. I've imported the ANDs, I've imported PD, NumPy, all the data set, all the libraries I've imported. Then I've specified the location of the data set I want to look at. For now, we'll be looking at the sub ID 002 itself, not all the 161. After defining the location, then we'll move on to Atlas. Uh, so let me explain the image, the atlas, how it is hitting RAM. So first, this path of our subject 2 we have stored. Then we will read the image and store it in T1. These are called T1 weighted images. So we'll store it in T1. Then we'll fetch the atlas, Harvard Oxford Atlas. We'll get this atlas. We'll store this atlas. Uh, then the same atlas is again read by ants and the atlas image is stored. So now we have T1 image, the original MRI image, then we have the atlas image. Then moving forward, what we'll do is we'll do the affine registration. So in this, we want to transform it using affine. We'll either scale it, we will crop it. We'll use these transformations, right? And we'll register. We'll register means nothing but putting labels to different regions of the brain. So we'll do that. This is the code for it. Now, Atlas in subject. This is our changed MRI. This is MRI which is perfectly labeled. Then we'll store this MRI over here. If I reload the output, see it gets stored. Subject to Atlas affine. So, like I said, the logic is very simple. Um, ants help us to register the brain. For registering, we make use of atlas. So ants help us to wrap around it. And affine is a type of transformation. It nothing but crop something out, expand something out, rotate something out, so that this atlas completely fits into our original MRI, and then we store it, as you can see over here. So basically, the offline registration is complete and safe. Then this is the exact code snippet I showed you about the Atlas labels and the different label IDs. Like I said, we've been making use of 11 and 21. We calculated the left volume, the right volume, and the asymmetric index using this very small formula, and we got this. So now we have understood this for one subject. We'll move on to understand it for the 161 subjects. It's nothing, it's simple, it is using for loop. So this is the exact same thing. I have taken the atlas, I fetched the atlas, and I've read the atlas. Then for my subjects, I'll run a loop for to store all the 161. I'll run a loop. This is the loop. Then I'll print the T1 path, the path in the NAT folder containing a subject ID and taking that specific MRI. And then we'll register it. We'll register it first, we'll leave the T1 image, and then we'll perform all the registration, and then we'll finally store it in a particular output folder. And after 2-3 hours, finally everything gets registered and the results are saved. Again, we'll calculate the asymmetry. Same thing, we'll run a loop to calculate the left wall, the right wall, then the AI. To calculate the Volex volume, this is the specific code for this. It is np.product. Product of what? T1 dot spacing. What is T1 dot spacing? So like I said, T1 was our original MRI image. Each MRI image has predefined length, breadth, and height. Those are known as spacings. So we do a product of the length, breadth, and height, and we get the Volex volume. And finally, yes, we store this in an Excel file. We call it results. It has the participant ID, it has the left volume, right volume, and the asymmetric index. You can see over here. Then we'll merge this with the participant folder like I showed you before. So if I've not showed the participant folder, it is in here. That is the participant.tsv. This is nothing but like an Excel file. It has all the participants. It has characteristics like can be group, it can be sex, age, education year, all the data is stored. For our purpose, we'll be seeing group. So what is this group? Every TSV file comes with a JSON. JSON is like a description, you can say. This description stated that group 1 means HC, healthy control. 2 means cocaine use disorder patient, CUD. So 1 and 2 are given. So using that JSON, the participants JSON, we'll merge it with our results. We'll merge it and we'll create a merged folder. So our merged Excel sheet is ready. In this, we'll be performing very simple analysis using matlab.plot, basic analysis. We can calculate the mean of both the groups. As we can see very clearly that group 1, the healthy control, has more leftward asymmetry, which is normal. But the 2, the CUD group, the addicted patients, they have less symmetry. They have less leftward symmetry. So that is very clearly visible. And we can even plot all the 161 patients and we get a curve like this. You can see that yes, it is skewed. The means are not equal. And yeah, that is basically the code. We can see that the Enigma addiction working group provided results that were very helpful. The mean itself, we can see group 1 has 6 and group 2 is 2, which is half the mean itself, which is a big thing. So let us now move on to conclusion. Conclusion simply states that the NACC asymmetric index for the healthy control was 6, around 6, and for the CUD was 3.5. This indicated that the CUD group showed reduced leftward symmetry corresponding to our research paper. The two sample had a t-test that showed 1.24. It indicates that this was not a statistically significant outcome. The difference in mean was not statistically significant. It is only because of the unavailability of very large data sets. 161 is a very small data set to provide any analysis with, but this is the only open neuro data set with a lot of amount of participants that I could find, but it does still match with the Enigma's research conclusion. The density plot also visually supports this, and that finally concludes this project. Thank you.